various category we are going to discuss, like carbohydrate, proteins, nucleic acids. So first we are going to discuss about carbohydrate. So what is a carbohydrate? The definition of carbohydrate is the carbohydrates may be defined as optically active polyhydroxy aldehydes or ketone. It means it is having more than one hydroxy. It means it is having more than one alcoholic group, and it can be aldehyde or it can be having the ketone group or the compound which produce such units on hydrolysis. It means the compound on hydrolysis which produce a unit which is polyhydroxyaldehyde or ketone. It is also known as carbohydrate. So we have to remember the term polyhydroxyaldehyde and ketone. Next, the most common sugar used in our home is named as sucrose, whereas the sugar present in the milk is known as lactose. We can say that carbohydrates are also known as saccharides. So how we can classify carbohydrate? So first of all, we will classify them on their behavior on hydrolysis. So first one is monosaccharide, second is oligosaccharide, third one is polysaccharide. So on the basis of their behavior on hydrolysis, it means how many unit they are going to produce on hydrolysis. First is monosaccharides. So what are monosaccharides? A carbohydrate that cannot be hydrolyzed further to give simpler units of polyhydroxy, aldehyde or ketone is called monosaccharide. So they cannot be further hydrolyzed. This we have to remember. They cannot be further hydrolyzed to give simpler unit of polyhydroxy, aldehyde or ketone. So these are known as monosaccharides. So how many monosaccharides are known in the nature? About 20 monosaccharides are known to occur in the nature. Some common example of monosaccharide are glucose, fructose, ribose, etc. These are the common example of monosaccharide. Now we are going to discuss the second category, oligosaccharide. So what are an oligosaccharide? These are the carbohydrate which yield 2 to 10 monosaccharide units on hydrolysis. So these are known as oligosaccharides. They can further be classified as disaccharide, trisaccharide, tetrasaccharide, etc. depending on the number of monosaccharides they provide on hydrolysis. So how many number of monosaccharide carbohydrate will produce? Based on that, we can categorize it into disaccharides, trisaccharides, tetrasaccharides, etc. Now, amongst these, the most common disaccharide, the two monosaccharide unit obtained on hydrolysis of a disaccharide may be same or different. The most common oligosaccharide are disaccharide. Disaccharide means it will produce two monosaccharide units on hydrolysis, and these units can be same or it may be different. Now we can see some disaccharide examples, and upon hydrolysis, what the monosaccharide they are going to produce. So sucrose, it will produce glucose and fructose You can revise or remember this by a simple trick, like we can learn it by SGF. Okay, SGF you can learn. So SGF, sucrose give glucose and fructose. Then maltose. maltose Maltose, you can see in maltose, it will going to produce two monosaccharide which are same, glucose, and second one is also glucose. So you can remember this because sometimes we get confused in that. So how we can remember? The starting letter is ma. Okay, so ma, it we can say that माँ के लिए दोनों हर बच्चा बराबर होता है ना, तो we can say that same type of monosaccharide will be produced. Okay, like that you can remember. Then lactose will produce glucose and galactose. Okay, next we are going to see polysaccharides. These are the carbohydrates which yield large number of monosaccharide on hydrolysis. Some common examples are starch, cellulose, glycogen, gums, etc. Polysaccharides are not sweet in taste, so hence they are also called as non-sugars. So one more important term is coming. The topic is reducing and non-reducing sugar. Previously, we have discussed about the hydrolysis product. So that can be asked in the MCQs. The hydrolysis product, it means the question will be like that, sucrose, what are the hydrolysis product for sucrose? So they will give in options, different options they will give. So you have to select the correct one. Reducing and non-reducing sugar can also come in MCQs. So what are reducing sugar? All the carbohydrate which contain aldehydic or ketonic group and reduce tolerance reagent or Fehling solution are called reducing sugar. Example, all monosaccharide, maltose, lactose, all monosaccharides, and then maltose, lactose, these are reducing sugar. Disaccharides such as sucrose, which give the two monosaccharide unit linked through their reducing center, they cannot reduce tolerance reagent or Fehling solution. So these are known as, this is known as non-reducing sugar. So you can remember sucrose is non-reducing sugar, and all monosaccharides and maltose and lactose are reducing sugar. So you have to remember the example in this. They can be asked in MCQs. Now we are going to see classification of monosaccharide on the basis of number of carbon atom and the functional group present in them. If monosaccharide contain an aldehyde group, it is known as aldose. And if a monosaccharide contain keto group, it is known as ketose. So let's see some example for that. Like if a monosaccharide having 3 carbon atom, the name given to it is triose. If it has aldehyde group, functional group, then it will be aldotriose. And if it is having ketone group, then it is known as ketotriose. Next, if the number of carbon atom is 4, then the general name given is tetrose. Then if it is an aldehyde, it is known as aldotetrose. If it is a ketone, it is known as ketotetrose. If the number of carbon is 5, it is known as pentose. If it have aldehyde group, it known as aldopentose. If it contain ketone group, then it is ketopentose. If the number of carbon are 6, then it is known as hexose. And if it is having aldehyde group, then it is known as aldohexose. And if it is having ketone functional group, then it is known as ketohexose. One more thing, in oligosaccharide on hydrolysis they give two monosaccharide, like disaccharide if we are taking, it will give two monosaccharide. How the monosaccharide are linked? These are linked by glycosidic linkage. This can also be asked in the MCQs, which type of linkage is present in them. Next, if the carbon number is 7, then it is known as haptose. If it is having aldehyde group, it is known as aldohaptose. And if it is having ketone functional group, then it is known as ketohaptose. Now we are going to discuss glucose in detail. First of all, we are going to discuss preparation of glucose. The first method of preparation of glucose is from sucrose. So if sucrose is boiled with dilute HCl or H2SO4 in alcoholic solution, glucose, fructose are obtained in equal amount. So this is the equation for that. So this is sucrose, it will give glucose and fructose. Then the next method is, so this can be remembered by SGF. Okay, so this can be remembered SGF. S stands for sucrose, G stands for glucose, and F stands for fructose. The second method is from starch. Commercially, glucose is obtained by the hydrolysis of starch by boiling it with dilute H2SO4 at 398 Kelvin under pressure. So what will the reaction in this? So here the reaction is given. If it is hydrolysis, then it will give, the starch is hydrolysis, it will give number of, N stand for number of, because if number is not fixed. Starch or cellulose give glucose. Then chemical properties of glucose. It is very important topic, and every time the question is coming in CBSE from this topic, either in the form of MCQs, either in the form of 2 marks or 3 marks question. So I request all the students to please practice these reactions, because this is the most important part of this glucose. So first one, glucose in an aldohexose, it means it contains 6 carbon and it is having the aldehyde group as a functional group. It is also known as dextrose. It was assigned the structure given below on the basis of following evidence. So how the structure of glucose is assigned to it? So various reactions were carried out. On the basis of that, the structure was given. So first of all, the molecular formula for glucose is C6H12O6. Now we will see the first reaction. On prolonged heating with HI, it formed N-hexane, suggesting that all the 6 carbon atoms are linked in a straight chain. So how we can say that all the carbon atom in glucose present in straight chain? By how we can say the evidence for this is, when we heat it with HI, then it will form N-hexane. N-hexane, this is the N-hexane, it is forming. So this will give the evidence that all the carbon atom present in the glucose are present in the straight chain. So we can remember glucose treated with HI, it will give N-hexane. Okay. So this will prove all the 6 carbon atom linked in a straight chain. So what we have to remember? We have to remember with which reagent we have to react and what will the evidence for that, what we are going to conclude from this reaction. The two things we have to remember. Next, chemical properties, reaction with hydroxylamine and hydrogen cyanide. So glucose react with hydroxylamine to form oxim. So this is the reaction of hydroxylamine and this will going to form the oxim here. And also glucose when react with hydrogen cyanide, it will form a addition product cyanohydrin. So this is the cyanohydrin. This complete molecule is cyanohydrin. Here the addition is taking place on this one. So this is the formation of cyanohydrin. So what these going to suggest, what they will confirm? They will confirm, these two reactions will confirm the presence of carbonyl group, that is CO in glucose. So the two reaction, what are the two reaction? Reaction of glucose with hydroxylamine to give oxim and reaction of glucose with hydrogen cyanide to give cyanohydrin. These two reactions confirm the presence of carbonyl group. So we have to remember the reaction and the confirmation, what they will going to confirm. Next chemical property is reaction with bromine water. So glucose get oxidized to 6 carbon carboxylic acid. It is known as gluconic acid on reaction with a mild oxidizing agent like bromine water. It will indicate that the carbonyl group present is an aldehydic group. So when glucose is treated with bromine water is a mild oxidizing agent. And what we are going to get the product? We are going to get the product gluconic acid. This is the gluconic acid. So what it will indicate? It will indicate that carbonyl group is present. That already in the second reaction we have seen. The carbonyl group which is present, it is aldehydic group. So it will confirm the aldehyde group, because carbonyl group can be of two types. It can be a ketone or it can be a aldehyde. So in this reaction with bromine water, it will confirm the aldehydic group. Now the next property, reaction with acetic anhydride. So in this reaction, acetylation of glucose with acetic anhydride give glucose pentaacetate. The product we are going to get the glucose pentaacetate, which confirm the presence of 5-OH group. Since it exists as a stable compound, 5-OH group should be attached to different carbon atom. So what will be the reaction in this? So what we have to remember, acetylation of glucose. It means we are treating it with acetic anhydride and we are going to get the product glucose pentaacetate. What it is going to confirm? It will going to confirm that it is having the 5-OH group are present in the glucose, and these 5-OH group should be attached to different carbon because it is a stable compound. The next property is reaction with nitric acid. On oxidation with nitric acid, glucose as well as gluconic acid. We have already seen gluconic acid, which was produced when we react glucose with bromine water, which is a mild oxidizing agent. So here, if we treat glucose as well as gluconic acid with nitric acid, both will yield dicarboxylic acid, that is known as saccharic acid. What it indicate? It indicate the presence of primary alcoholic OH group in glucose. So the reaction is like this. So this is glucose, and when it treat with, we do is its oxidation with nitric acid, and gluconic acid oxidation with nitric acid, we take nitric acid, both will give the product saccharic acid. You can see the difference. When we are taking glucose, both this carbon and this carbon, they change into COOH. And here, this is already COH, this will change into COOH. So both will give the saccharic acid. And what it will indicate? It will indicate the presence of primary alcoholic OH group in the glucose. So it will give the presence of primary OH group in glucose. Next, we are going to discuss the open chain structure of glucose. From above chemical properties, what we have already discussed in our previous slides, it is clear that glucose has one aldehyde group, one primary alcohol group. Since glucose have 5 OH group, so 4 OH group must be secondary because one is primary. So from the above point, the possible open chain structure of the glucose should be, it can be like this. The exact spatial arrangement of different OH group was given by Fischer after studying many other properties. Its configuration is correctly represented as the structure which is first. This is the Fischer projection of glucose. Here the horizontal line represent the it will represent the bond is toward the observer, and the vertical line represent the bond is away from the observer. So this is the Fischer structure of glucose. Next is gluconic acid, and other is saccharic acid. These three Fischer structures are given after the studying all the properties which we have already discussed in our previous slides. Here you can see that aldehyde group is present, 5 OH group is also present, all these carbon are in straight chain, one primary OH group is also present. Next, we are going to discuss D and L structure of glucose. Glucose is correctly named as D plus glucose. So here, capital D, we have to see it is capital D, not small d. Small d represent dextrorotatory, or it can be represented by plus sign. But here the symbol is given capital D. So what is the meaning of using this symbol capital D for this? So glucose is correctly named as D plus glucose. D before the name of the glucose represent the configuration, whereas positive represent dextrorotatory nature of the molecule. So here capital D is we are using in reference as a reference molecule. We are taking glyceraldehyde. With its reference, we can say that this is D or L form. So glyceraldehyde contain one asymmetric carbon atom and exist in two enantiomeric form. So here we can say that this is the two form of glyceraldehyde. And this is our glucose. We have to see this one. I use the dash line here. The box is given. Now see, the OH group is on the right hand side. They are having the same configuration. So this is D-glyceraldehyde. This one is D-glyceraldehyde, and if the OH is on left, then it is capital L we are using. So this is resembling this structure. So that is why we use D plus glucose with reference of this molecule. Now, what are the objections for the open chain structure of glucose? So from the chemical properties which we have already discussed, we come to know that we conclude that it is having a open chain structure, but in that also there are some objections. So this is also very important part. So इसमें we have to see first one, what is that? Despite having the aldehyde group, glucose does not give Schiff's test. It means Schiff's test is when we add Schiff's, we take Schiff's reagent and we add, it is colorless. When we add any aldehyde compound containing aldehyde group, then its color will change to magenta. So it will not give the Schiff's test. It does not form the addition product, hydrogen sulfide addition product with NaHSO3. Next one is the pentaacetate of glucose does not react with hydroxylamine. Indicate the absence of free CHO group. It means in the open chain structure, the CHO group is shown in free form, but if it is in free form, it should give this test that pentaacetate glucose should react with hydroxylamine, but it does not react because it does not contain free CHO group. This will discuss later on. Glucose is found to exist in two different crystalline form named as alpha and beta. A stand for alpha, B stand for beta. The alpha form of glucose, its melting point is 419 Kelvin and it is obtained by crystallization from concentrated solution of glucose, while the beta form, its melting point is 423 Kelvin and is obtained by crystallization from hot and saturated aqueous solution at 371 Kelvin. So glucose exists in two form, alpha and beta form. This behavior could not explain by open chain structure. So you have to remember these points. These show that glucose does not have open chain structure because the open chain structure does not describe these points. Now we have to discuss the cyclic structure of glucose. It is also known as Haworth structure. It was proposed that one of the OH group may add to the CHO group and form a cyclic hemiacetal structure. So we can see that it was found that glucose form a 6-member ring in which OH at C5 involved in the ring formation. This one, this OH is involved with this carbon in ring formation. This explains the absence of CHO group. So the structures can be like this. So here this one is the alpha form, alpha D glucose, and this is beta D glucose. How we can say that it is alpha and beta? Based on the configuration of on C1 carbon. If it is on C1 carbon, OH on the right side, then it is alpha. And if it is on left side, it is beta based on the configuration. So here the CHO group is involving in the ring formation with the OH group of 5th carbon. So that is why it is not free, and this ring formation, this structure will explain the absence of free CHO group. Now in the cyclic structure of glucose, the 6-member cyclic structure of glucose is called pyranose because it is derived from the ring pyran, analogy with the pyran. So how we can draw this, you can see. Like this we can draw the structure. This structure is very important. It is already asked in previous year CBSE board questions. So in this, what we have to remember, this is carbon 1, this is carbon 2, this is 3, this is 4, and this one 5, and here the 6th one will come. Now what we have to see, like this you can draw. After that you have to see the group which is present on the right hand side will be, the group which is present on the right hand side will below, we have to write here, and the group which is on left hand side, that this will we have to write on above the line like this. In the first carbon you can see left H and right OH. So left will written on the top and on the bottom we have to write the OH. So what the group on the right hand side, that will we have to write on the bottom, and on the left side that we have to write on the top. Like same, we can write this left side H, right side OH. So we can write it like this. Then you can see in the third carbon we have seen OH is on the left. So it will write on the top of the line, and H is written on the bottom of the line. Next, we have seen H on the left hand side. Above we have to write, and below we have to write the OH. Likewise, you can see. It is easy to, if you use this trick, you can easily remember what we have to write on the bottom and what we have to write on the top of the line. Then again, H and CH2OH. This is a 6 carbon, this one. So that is like this you can draw the Haworth structure or cyclic structure of glucose. This is alpha D glucose, because here on the C1 carbon, the OH is present on the right hand side. So it is alpha D glucose. The 6-member cyclic structure of glucose is called pyranose, and it is in two form. We will have alpha and beta form, because it resemble analogy with, it is having analogy with pyran ring. So now we are going to see beta D glucose. So likewise, same we have to draw. Only we have to see the configuration at C1 carbon. We have to change all the things on all other group will be as it is. Only the configuration at C1 carbon we have to change. There in the alpha form we have, on the top we are having H, and on the bottom we are having the OH, but in the beta form we have OH on the top and H on the bottom. Only this will change, all other groups will be same. So these alpha and beta form are known as anomers. So what are anomers? The compounds which differ from each other only at C1 configuration. Now we will see fructose. It was found to contain a ketonic functional group. So remember, in glucose we have seen glucose is having aldehyde group, but fructose is having ketonic functional group at carbon number 2. It is also 6 carbon straight chain. It belongs to D series. It is Leo rotatory compound, so we will write it like this, D minus fructose. Let us see the structure. So this is the straight line structure, open chain structure of fructose. Here also, like glucose, the carbons, the OH group is involved in the ring formation. Here also we are having the two forms. This is this one. This is the alpha form, and this form is the beta form. Sorry, this is not the alpha form, this is the alpha form. This is the fructose. Okay, after ring formation it is giving alpha form and beta form. So here in this form you can see here OH on the right hand side, the configuration here on C2 carbon OH, and here it is on left hand side. So this is the two form of the fructose we are having. My voice is audible to everyone? Is my voice is audible to everyone?
Yes, audible, madam. Audible. Audible. Please go ahead.
Thank you, sir. Now, oh, thank you, sir. Thank you, students, for your response. Now we are going to see the cyclic structure of fructose. This is the cyclic structure of fructose. This is alpha D fructose, and this is beta D fructose. So here we can see the configuration here change only is at this carbon, second carbon. So this is also, the fructose is also exist in two form, alpha D minus fructofuranose, because it is derived or it is having the analogy with the furan ring. And this is beta D minus fructofuranose. This is the complete name for fructose. Now we are going to discuss amino acids. Amino acids, what are the groups which are present in amino acids? First is it is having NH2 group and amino group, and second it is having the carboxylic COH functional group. Depending upon the relative position of amino group and with respect to carboxylic group, the amino group can be classified as alpha, beta, gamma, delta, so on. Only alpha amino acid are obtained on hydrolysis of protein. They may contain other functional group also. So amino acid are usually colorless crystalline solid. These are water soluble, high melting solids. Amino acid are generally represented by a 3-letter symbol. Sometimes 1-letter symbol is also used. Structure of some commonly occurring amino acid along with their 3-letter and 1-letter symbol are given. Now we will see the table which is from NCERT. So these are the amino acids which are given. Here the structure is not asked in the previous year, only they can ask you the essential, non-essential amino acid, they can ask. So you have to remember the name and the definition for that. So the amino acid which are having the star on them, these are the essential amino acid. So what are essential amino acid? Essential amino acid are the amino acid which are not synthesized in our body. So we require it from outside, and the non-essential amino acid are which our body will synthesize, which is synthesized in our body, that are non-essential amino acid. So this is given star. If it is having the star, it is essential. I will give a trick to you to remember the essential amino acid. These are some more amino acid. Here you can see this isoleucine, arginine, lysine, threonine. Here some more amino acids are given, like methionine, phenylalanine, tryptophan, histidine. So you can remember the essential amino acid by a trick, which is we can say that VIP Hall. It is not written properly. You can write it. I will tell you. VIP Hall में TT. In that you can say from the first table VIP. In this VIP, V is valine, then I is isoleucine, P is phenylalanine. Then VIP Hall HALL. In that H is histidine, A is arginine. They are given in table. It is in arginine. L is lysine, and one more leucine. VIP Hall में methionine, ME में से we can get methionine, and then we will have TT. VIP Hall में TT. TT means one T stand for threonine and other T stand for tryptophan. So this is VIP Hall में TT. So this is the trick to remember the essential amino acids. Now we will see the classification of amino acids. On the basis of their nature, if the number of amino acid, if the number of amino and carboxylic group are equal, it is neutral. If the number of amino group are more than carboxylic group, it is basic. If the number of carboxylic group are more than amino groups, it is acidic. On the basis of occurrence, how we can classify amino acid? Amino acids which can be synthesized in our body, that this already I have discussed with you. If it is synthesized in our body, these are known as non-essential amino acid. The definition can be come in 1 marks. And which cannot be synthesized in our body are known as essential amino acid. The definition can become, so we have to remember the definition. Now what is a zwitterion? Amino acid behave like a salt rather than simple amine or carboxylic acid. This behavior is due to the presence of acidic carboxylic group as well and the basic amino group in the same molecule. In aqueous solution, the carboxylic group can lose a proton and amino group can accept a proton, giving rise a dipolar ion. It means both positive and negative charge are present in a single ion. So it is known as zwitterion. It is also a very important question, zwitterion. It is asked in 1 or 2 marks question. So what is zwitterion? In aqueous solution, the carboxylic group of amino acid lose a proton and the amino group will accept the proton. So by result we will get a dipolar ion which is known as zwitterion. This is neutral but contain positive and negative charges. In zwitterionic form, amino acids show amphoteric behavior. As we know, what is an amphoteric behavior? Amphoteric behavior means the compound, the ion will react with the both acid and the bases. Now we can see in aqueous solution what is the changes take place. This H from the carboxylic, it will accepted by this base amino group. So it will become NH3 positive and it will become O minus. So this is the zwitterion. This is the zwitterion form. It is an important question. You should remember this one also. Then peptide linkage. So we have discussed in carbohydrate how the monosaccharide are connected with each other. They are connected with each other by glycosidic linkage. In amino acids, in proteins, in amino acids, these amino acids are connected with each other by peptide linkage. So when the number of amino acid more than 10, then 10 are bonded, the product are called polypeptide. Peptide linkage, what is peptide linkage? It is an amide linkage which is formed from between COH group and NH2 group of other amino acid. If 2, 3, 4, 5, 6 amino acid are bonded, then product are dipeptide, tetrapeptide, pentapeptide, as like that. And when the number of amino acid more than 10, then the product is known as polypeptide. We have to remember these linkage name. In which type of biomolecule, which type of linkage is present. In carbohydrate, we have seen the linkage was glycosidic, and in protein amino acid, we have seen peptide linkage is present. Now, what is a protein? A polypeptide with more than 100 amino acid residue having molecular mass higher than 10,000 U is called as protein. Now we have to see the classification of protein. On the basis of their molecular shape, First is fibrous protein. What is a fibrous protein? When the polypeptide chain run parallel and are held together by hydrogen sulfide, hydrogen bond and disulfide bond, then fiber-like structure is formed. Such protein are known as fibrous protein, and these are insoluble in water. Some example for them are keratin, which is present in our hair, wool and silk, and myosin, which is present in our muscles. So the first category on the basis of their molecular shape, it is fibrous protein. Next is globular protein. What is a globular protein? This structure results when the chain of polypeptide coiled around to give a spherical shape. These are usually soluble in water. So fibrous protein are not soluble in water. It means they are insoluble, and globular protein are soluble in water. Example for globular protein are insulin and albumin. These are some common examples. Now these are the classification of protein. Now we are going to discuss the structure of protein. So protein is having some protein is having primary structure, some secondary, then tertiary, then quaternary. This type of structure shown by the protein. So what is a primary structure? Primary structure, protein may have one or more polypeptide chain. Each polypeptide chain is a protein and has amino acid linked with each other in a specific sequence. It is the sequence of amino acid that is said to be primary structure of the protein. Any change in the primary structure or in the sequence of protein create a different protein. It means a specific sequence is there, and these amino acids are linked by peptide linkage. So they are forming the primary structure. What is a secondary structure? The conformation of polypeptide chains assumed as a result of hydrogen bonding is called as secondary structure of protein. It means in this we are having some more bond in addition to the peptide linkage. Hydrogen bonds will also form, so which give rise to secondary structure of protein. Depending upon the size of R group, two different structure are possible. First is alpha helix structure and second is beta pleated structure. This is the alpha helix structure. The second one is the beta plated structure. So here, when you twist a rope or anything, it will give you a secondary structure, which is alpha helix structure. And when these type of structure join together, interlinked with each other by hydrogen bonding, then these known as alpha. This will form a sheet-like structure. This is form a sheet-like structure. So this is known as beta plated structure. Now what is a tertiary structure? Tertiary structure of protein represent overall folding of polypeptide chain. So now the folding will start of secondary structure. It gives rise to two major molecular shape, can be fibrous or globular. The main force which stabilize the secondary and tertiary structure of protein are hydrogen bonds, disulfide linkage, van der Waals force, and electrostatic attraction. The last one is quaternary structure. Some of the proteins are composed of two or more polypeptide chain referred to as subunits. So in quaternary structure, we are having two or more polypeptide chain. The spatial arrangement of these subunit with respect to each other is known as quaternary structure. We can see in the diagram also. This is the primary structure. In this, the different amino acid. These are the amino acid, and these are linked together by linked to each other by peptide linkage. Then some twisting will take place in the polypeptide chain, and these twisting will link with each other with the help of hydrogen bonding like this. After that, coiling will start in the secondary structure. It is known as tertiary structure. Then if the coiling is there and different polypeptide chain coiling is there, one means it is containing two polypeptide chains, so coiling in one and then other. So this one is one polypeptide chain and this is the second one. So this form a quaternary structure. Now it is also a very important question, denaturation of protein. It is asked in previous year so many times, so you have to remember this and learn it. That is denaturation of protein. So what is denaturation of protein? See, protein found in a biological system with a unique three-dimensional structure and biological activity. This is known as native protein. When a protein in its native form is subjected to physical change like temperature or chemical change like in pH, the hydrogen bond will disturb. Due to this, the globules unfold and helix get uncoiled and protein lose its biological activity. So, by subjected to any physical change or chemical change. In physical, we can say that change of temperature. In chemical, we can say the change in the pH or in hydrogen bond. So, after this change, the hydrogen bond will disturb and the protein will lose its activity, biological activity. So it is known as denaturation of protein. So it is a very important question. So during denaturation, what happened? During denaturation, secondary and tertiary structure are destroyed, but primary structure remain unaffected. So it means the secondary and tertiary structure will be converted or destroyed and form the primary structure, and primary structure will not be affected by denaturation. Now we can see some example. The coagulation of egg white or on boiling, and the curdling of milk, which is caused due to the formation of lactic acid by the bacteria present in milk. These are the example of denaturation of protein. Now we are going to discuss chemical composition of nucleic acid. So complete hydrolysis of DNA or RNA yield pentose sugar, phosphoric acid and nitrogen base. Pentose sugar. So in RNA, the pentose sugar present is beta D ribose. And in DNA, the pentose sugar present is beta D 2-deoxyribose. Here you can see at carbon number 2, here the group is OH and here only H is present. So the name is beta D 2-deoxyribose, and in RNA it is beta D ribose. One oxygen is not there in the second position. Now nitrogenous bases. There are two type of nitrogenous bases, purines and pyrimidines. No need to remember the structure for these. Only name you have to remember. What are purines? Purines are adenine and guanine. Pyrimidines are cytosine and thymine. These are the nitrogenous base which are present in the DNA, RNA. And one more is there, uracil. I will tell you the difference in RNA and DNA later on. Now we can see nucleoside and nucleotide. It is also an important question. They can ask you the difference between, what is the difference between nucleosides and nucleotides. So what is nucleoside? See, we can use a simple trick here. See, sides. Road is having two sides. It means nucleoside is having two component, nitrogenous base and pentose sugar. And this is the structure for that, the pentose sugar with a base. It is a nucleoside. Then nucleotide, it is having three component. Tides or tri you can use. It tri means three. So it is having three component, nitrogenous base, pentose sugar and phosphate group. So side you can remember road is having two sides, so it contains two component, nitrogenous base and pentose sugar. Then tides you can use tri, so it is having three component, nitrogenous base, pentose sugar and phosphate group. So this is the structure of the nucleotide. This is the nucleotide. Now how the nucleotides are formed? So two units will combine with each other with the help of phosphodiester linkage. So you have to remember two nucleotide, these will combine with each other with the help of phosphodiester linkage. Remember, in carbohydrate we have discussed glycosidic linkage. In proteins we have discussed amino acid, we have discussed peptide linkage. In RNA, DNA, nucleotides, we are going to discuss the phosphodiester linkage. So here the fifth carbon, this one, 5 dash is attached to the 3 dash of the other nucleotide. So this will, they will link and they will form the chain like this. So this is the structure of DNA. So this structure is given by James Watson and Francis Crick, gave a double-stranded helix structure for DNA. Two nucleic acid chain are wound about each other and held together by hydrogen bond between pairs of bases. The two strands are complementary. These are complementary to each other because the hydrogen bond formed between specific pair of bases. So this we have to see. Adenine form hydrogen bond with thymine and cytosine with guanine. This you have to remember. This is the Watson-Crick double helical structure. Then structure of RNA. In secondary structure of RNA, a single stranded helix is present, which is sometimes folds back on itself. RNA molecule are of three types. They form different functions. They perform different functions. They name as messenger RNA, mRNA we usually call it, then ribosomal RNA, rRNA, then transfer RNA, tRNA. These are some type of RNA. Then DNA fingerprinting. So a sequence of bases on DNA is also unique for a person. And information regarding this is called DNA fingerprinting. Application, what are the application? In forensic lab for identification of criminals, like we have already heard about that DNA test. The police and they do the DNA test that for identification of criminals to determine the paternity of a person. If any issue is there, to find that, we use the DNA fingerprinting. Then to identify the dead bodies in any accident, comparing the DNAs of their parents or children. So if the dead body is damaged so much that it cannot be identified, so we use DNA fingerprinting for that. Next, to identify racial groups to rewrite the biological evolution. So in that also we are using the DNA fingerprinting. Then this question also asks difference between DNA and RNA. First one is the sugar present in DNA is deoxyribose sugar, and in RNA it is ribose sugar. This is oxyribose sugar, and this one is deoxyribose, because at second position the oxygen was not there. Here the oxygen was this. They can say at ribose sugar also. So second point is DNA contains cytosine, thymine as pyrimidine base, and in RNA cytosine, and in place of thymine it is uracil is there. So this is the change. Third one, DNA has double stranded alpha helix structure and RNA is having single stranded alpha helix structure. Then DNA has unique property of replication. RNA usually does not replicate. DNA control the transmission of hereditary effect, and RNA control the synthesis of proteins. These are the difference between RNA and DNA. So the chapter biomolecule is completed. Now I am going to discuss some reasoning question of inorganic chemistry.
मैम टाइम नहीं हुआ है
not, I'm not switching off. I'm just discussing important question of inorganic chemistry. I'm not closing. Wait a second, give me. Now, these questions are visible to you. Are you able to see the questions? Are you able to see the questions?
Yes, visible, madam. Visible, visible.
Okay, thank you, sir. Now we are going to discuss some reasoning questions, some important question of inorganic chemistry. Yesterday also I have discussed some questions. Now also I am going to discuss this. The first question we are going to see, which property of transition metal enable them to behave as catalyst. So I have already discussed with you. Wait a second. So here we see which I will again just read the question. Which property of transition metal enable them to behave as catalyst? So what was that? This is the what are the options? First is high melting point, second is high ionization enthalpy, and third is alloy formation, and D is variable oxidation state. So they are behaving as a catalyst due to variable oxidation state. So this is the reason for that why D block element transition metals behave as catalyst. Next, we are going to see the interstitial compound of transition metals are soft. So they are asking about the properties of interstitial compound. So in that, one is softer, A is softer, B is more ductile, C is harder, and D is more metallic. So the correct answer will be harder. So this was the property which was given in the properties of interstitial compound. Next, which of the following characteristic of transition metals is associated with the catalytic activity? This is the same question as I have discussed with you. Variable oxidation state is the answer. Here the options are changing, but the answer will remain the same. For catalytic activity, variable oxidation state is the reason. Then which of the following transition metals show +1 and +2 oxidation state? Here in this, +1 and +2 is shown by copper. The option are given manganese, then zinc, then scandium, then copper. The answer will be copper. Copper is the answer, which is the transition metal which is showing the 2 oxidation state. Next question is, the ions of group 12 zinc, cadmium, mercury have completely filled d orbitals, so this question I have already discussed with you yesterday. So what we call them or what are the property they show? You can see first option is behave like semiconductor. No, we have not discussed this one. Or very high melting solids. No, then do not behave like transition metal, and D option is behave like superconductor. So the reason or the property which is because they are having completely filled d orbitals, so they do not behave as transition metal. So if they ask about group 12 element zinc, cadmium, mercury, they are having fully filled d orbital, so they do not behave as transition metals. Next question is, define transition metal. Why zinc, cadmium? The same question is asking. These all are previous year questions. The same question. First of all, what is transition metals? So we have already discussed yesterday the elements which are having either in their ground state or in their excited state, they have incompletely filled d orbital or partially filled d orbital. They are regarded as transition metal. So why zinc, cadmium, mercury are not regarded as transition metal? The reason for that is because they do not have incompletely filled d orbitals. All the orbitals are filled in the zinc, cadmium, and mercury. So they are not regarded as transition metals. Next question, why do transition metals show variable oxidation states? how are transition metal different from p block elements in terms of variability of oxidation state. So why do transition metals show variable oxidation state? This type of question I have already discussed with you in this, because there is a less energy difference between ns and n minus 1 d orbitals, because they are having comparable energy difference is less, so they are showing the variable oxidation state. So next part of the question is how transition metal different from p block element in terms of variability of oxidation state. So in p block element, the oxidation state is differ by 2, whereas in transition metal the oxidation state is differ by 1, like plus 2 plus 3 plus 4 plus 5, like that it will change, but in p block element it will change by 2 units. This is the difference. Now, if the question is asked account for the following, it means they are asking for the reason. So in this next question is Cr2 plus is strong reducing agent. Strong reducing agent means it will reduce the other species and it will get oxidized. So after oxidation, it will form Cr3 plus. So why it is forming the Cr3 plus? Because it is stable electronic configuration. We already discussed the when ligand approach any transition metal, then the d orbital will split up into T2G and Eg level. So in Cr3 plus, it is having half filled T2G level. So that is why it is stable. So it is a strong reducing agent. Now, of the D4 species, Cr2 plus is strong reducing, while Mn3 plus is strongly oxidizing. So we can see in previous here also in previous question also we have discussed Cr2 plus, because it will get the half filled T2G electronic configuration. So half filled configuration is more stable. That is why it is formed Cr3 plus, and it is a strong reducing. But if Mn3 here's they are telling Mn3 plus is strongly oxidizing, it means it will oxidize the other species and it will get reduced, and it will reduce Mn2 plus. So why it is getting easily reduced? Because after reduction it will get 3d5, the half filled orbital half filled d orbital it will get. This is more stable. Next question is the D1 configuration is very unstable. So what is the reason for that? These ions have low hydration enthalpy, therefore undergo disproportionation reaction to become stable, like Cu plus, it will change into Cu2 plus. Some more question we can discuss. Give reason for the following statement. Copper does not displace hydrogen from the acid. This we have seen in the electrode potential yesterday. Copper is not able to displace the acid because copper is having the positive reduction potential, or we can say that it is having positive standard electrode potential. Now, the next question is zinc, cadmium, and mercury are soft metals. So, what is the reason why they are soft metal? The softness or the metal melting point, these things depend on the metallic bond. So, zinc, cadmium, mercury, they are not having the unpaired electron. Though due to the absence of unpaired electron, and these unpaired electron are responsible formation of metallic bond. So if unpaired electron are not present, so the metallic bond are weak and they are soft. Which ion amongst the following is colorless and why? So in this we can see this type of question. These all are previous year questions. Ti4 ion is colorless. If we write the electronic configuration, we will see Ti4 plus is colorless because here unpaired electron are not present, and if unpaired electron is not present, then it will not undergo DD transition. We have seen yesterday, due to DD transition, we the species will show the color. Next is why Mn2 plus much more resistant than Fe2 plus toward oxidation. So here also we will see the electronic configuration. So Mn2 plus is 3d5. It is more stable because it is half filled. But in Fe2 plus it is 3d6. It is not very stable. So that is why Mn2 plus is more resistant. The next question is silver atom has completely filled d orbital in its ground state. It is still considered to be a transition element. Justify. So they can ask the question in this way also, because in zinc, cadmium, mercury, they are asking the why they are not regarded as transition metal. Here they are asking why this is regarded as transition metal, because we have the condition for transition metal that either in their ground state or in their excited state or any ion form, they are having the incompletely filled d orbital. So silver is having filled d orbital in its ground state, but when it formed the Ag2 plus, it is having 4d9. So that is why it is having the incomplete d orbitals in Ag2 plus state. So that is why it regard as transition metal. Then why do transition metals form alloys? So what is the reason? I discussed with you yesterday. It is due to small atomic size. They are having same size, or almost same size. So smaller atomic size and same size, they are replace each other. So their atomic size is not much different from each other. So that is why they are forming the alloys. Next is following ions of 3D transition series are given. So few ions are given. Identify the ion which is. So in among these ions, the atomic number is already given. So we have to identify the most stable aqueous, most stable in aqueous solution. So here the Cr3 plus will be more stable because it will form half filled T2G level. Then a strong oxidizing agent. Strong oxidizing agent means it will oxidize the other species and itself get reduced. So here Mn3 plus will reduce to Mn2 plus to get the half filled orbital which is more stable. Then a colorless aqueous solution. Which species will give a colorless aqueous solution? So Ti4 plus species is giving the colorless aqueous solution due to the absence of unpaired electron. If unpaired electron is not present, no DD transition will take place. So the species will not show the color. Some more reasoning question we can also see in that. First question, manganese exhibit highest oxidation state of +7 among the series of transition element. What is the reason for that? Due to the presence of large number of unpaired electron. So it is having the it is having large number of, because the d orbital is half filled, so it is having more large number of unpaired electron, and it will form many take part in the bond formation. So it will show the highest oxidation state. Next question is zinc has lowest enthalpy of atomization. So why zinc has lowest enthalpy of atomization? Zinc has large size and 3d10, its electronic configuration is 3d10. Therefore, it form a weak metallic bond. Hence, it has a lowest enthalpy of atomization. Means it will require less energy to convert in vapor form or atomic form. Now, some reasoning question from some important compound of transition metal. First is give reason. E naught value of Mn3 plus slash Mn2 plus couple is much more positive than that of Fe3 plus 2. It will change into Fe2 plus. So what is the reason for this? It is because Mn2 plus is half filled, which is more stable as compared to Mn3 plus, which is having 3d4. And third ionization enthalpy is high. High energy is needed to convert Mn2 plus to Mn3 plus. Whereas Mn2 plus is 3d6 and it is less stable, and if it lose one electron, it will form 3d5, which is more stable. So that is why it is more positive as compared to Fe3 plus to Fe2 plus. Next question, calculate the number of unpaired electron in following gaseous species. So for that you you have to remember the atomic number, how to write the electronic configuration. So in this, the calculate the number of unpaired electron following gaseous ion, which the ions are given, which one is more stable and why. So in this Mn3 plus and Cr3 plus, which one is more stable? So here we can see that Mn3 plus is having 3d4, 4 unpaired electron, and Cr3 plus is having 3d3. But we can see that it is having T2G level half filled, so this is more stable. Now MnO is basic. This is also we have discussed yesterday also. MnO is basic, whereas Mn2O4 is acidic in nature. Give reason. So we have discussed that the acidic nature will increase with the increase in the oxidation number. So that is why it is MnO is ionic, therefore basic, and Mn2O7 is covalent, and that is it is acidic. Some more questions are also there. So here some E naught values are given for different elements, and some reason are asked here. So in the first one, which is stronger reducing agent in aqueous medium, Cr2 plus or Fe2 plus? Why? So Cr2 plus is stronger reducing agent because it will oxidize itself to Cr3 plus, which is having the half filled. See, you can see that same type of question are repeating again and again in different years and in different way. So the reason for that is same, but the pattern of asking the question is different. So you have to see the different way the same question is asking. And Fe2 plus is not a strong reducing agent because it will get 3d5 in Fe3 plus, but here T2G half filled. Next, we have say the elements of 3D series are given as we have to answer from the following. Which element has the highest melting point and why? So in that we have that chromium has the highest. We have seen the graph. So please study the graph properly, and So in that we have discussed chromium has the highest melting point due to large number of unpaired electron. In that magnesium and technetium, these are asked that why they are not having highest, because their electrons are not delocalized. Which element is more stable in plus 2 oxidation state and why? Mn2 plus is more stable because it is half-filled 3d orbital. Which element has lowest enthalpy of atomization and why? Zinc, we have already discussed. Zinc is having the lowest enthalpy of atomization because it's not having the unpaired electron. So it will not form the strong metallic bond. Then preparation question is asked. So please study the preparation of uh, K2Cr2O7 potassium dichromate, and so this is the preparation they are asking. So you have you have to revise this also. And KMNO4 also please study potassium permanganate preparation also. Then some more reason question we can see. Zinc 2 plus salt are colorless while Ni2 plus salt are colored. So what is the reason? Because Zn2 plus does not have unpaired electron, but Ni2 plus have unpaired electron. Then Cr2 plus is strong reducing agent. This we have already discussed. Same, because it is having T2G level half fill, is having T2G3. Transition metal and their compounds show catalytical activity. This also we have discussed because they are having variable oxidation state. Then transition metal form complex. Uh, why they are forming complex? So three reasons for that. They are forming coordination complex. Why? Because small size, high charge and vacant d orbital to accept the electron from the ligands. Then E naught value for Mn2 plus to Mn, and value for manganese is highly negative as compared, whereas for Mn3 plus slash Mn2 plus is highly positive. So what is the reason for this? Because it is because Mn3 plus 3d4 is unstable as compared to Mn2 plus. Then Cu plus ion is unstable in aqueous solution. So why? Because Cu2 plus get oxidized to Cu2 plus because they show disproportionation reaction. What is the reason? Because it is having higher enthalpy of hydration. Cu2 plus is having. Now again the question is for the preparation. I hope all of you like the class and understand the topic. Uh, Thank you all, and thank you for your cooperation.
Thank you, ma'am. Good night.